I wanted to ask you about hemophiliacs. Because they had cell-free plasma, it was just the virus. But the virus sheds its membranes within 24 hours. So one thing we couldn't get is how was it able to actually infect the T cells?
Yes, it's a question, but we have to know that all the fraction of the blood could be infectious. And there is some virus bound to red blood cells which could be released also in the plasma after treatment or incubations. Some virus is bound to red cells also as well. Perhaps there are more virus when you process the blood. More virus could come in the plasma. Okay, and these virus could be protected by the plasma proteins from denaturation. This is one thing, one possibility. The other is the quality of the host. Hemophiliacs are fragile. Being transfused many times, their immune system also is depressed before they get infected with HIV. So they are prone to HIV infection because of their immune system weakness. Okay. So combining this with the fact that the virus could exist also in forms which I'm studying now, which probably are more resistant than we think for the usual particles, could explain.
The little ones.
Yes, little ones, or they bound to mycoplasma proteins. You know, that's possible that mycoplasma are more resistant, perhaps. Mycoplasma envelope would be more resistant than the viral envelope.
Okay.
But there are all hypotheses. This is not based on solid data, of course. This is just they are just assumptions. But you are right. We have to explain why hemophiliacs have been so easily infected with plasma products.
For you, the entire existence of HIV rests upon the fact that there are no pictures of the purified gradient. Is that correct?
It is part of it. That is the most crucial evidence which you need. If you don't have these pictures which prove that there are, in the purified virus, what they call purified virus, they are virus-like particles, then the whole experiment and thus the existence of HIV is finished.
And you're saying to date there is no pictures of purified virus?
Today there is no pictures of purified virus. And certainly, Montagnier did not publish it, Gallo did not publish it, Levy did not publish such pictures, Weiss did not publish such pictures. And the only pictures which have been published was, in fact, this is admitted by the Franco-German researchers in 1997, when the first attempts, the first pictures of what is called purified HIV were published by two groups, one from the United States and one in a Franco-German study.
You said in 1997 they did try to purify HIV, is that correct?
Yes.
And you're saying they weren't successful?
They're not successful. And what is more important, these authors, they accepted or they admitted, but by 1997, there is no evidence for purification. And here it is. This is from the Franco-German study. Virus to be used for biochemical and serological analysis or as an immunogen, that is as an antigen, is frequently prepared by centrifugation through sucrose gradients. And they said, in none of these studies has the purity of the virus preparation been verified. So by 1997, there is no proof that HIV has been purified.
But did they purify HIV?
They tried. So they accepted this. As I said, we've been asking this for the very beginning to have some evidence for HIV purification. And these authors tried to present, or they did their best to purify HIV. And here is their evidence. This is the Franco-German study. And they had material which is meant to represent purified HIV. The top and the middle is material obtained from infected cultures and is meant to represent purified HIV. The bottom part is material obtained in a similar manner from non-infected cultures. And as you can see, the arrows, they said, represent the HIV particles. First of all, this cannot be said to be purified virus. As you can see, they are not purified particles. In fact, only the particles which they put the arrows are said to be particles which look like HIV. The rest are all cellular fragments, or called vesicles. In the material which is obtained from the non-infected cultures, you can see even there are some particles which may look like the one which is arrowed. So it is significant that the authors do not call this material purified HIV. In fact, they call it purified vesicles from infected H9 cells, the top, and activated cells. So the authors admit that this cannot be considered purified HIV. With all the effort they put, they could not obtain purified HIV. Okay. It is also important that the particles which are arrowed as representing HIV, they don't have all the morphological characteristics attributed to HIV. There is no evidence for knobs. In fact, even their diameter is higher than what is considered to be the retroviral particles.
But does size really matter? I mean, humans vary in sizes.
They matter because this in the Franco-German the size is, they are larger, but not as large as in the American evidence for purification. Here is the effort by the American researchers in purifying HIV. These are their results. And as you can see, they were not able to obtain purified HIV. And again, the particles which are arrowed and are said to represent HIV, they do not have all the morphological characteristics attributed to lentiviruses. And in fact, they don't have the knobs, they don't have cone-shaped core, you cannot see there, they don't have lateral bodies, which are, they should be in HIV, and most importantly, their diameter is they're too large. The average diameter of the particle is 234, and none has a diameter less than 160. Just by taking their diameter, it is impossible the particles which are labeled as HIV to be HIV.
Is the size also important in determining what you're looking at?
Yeah. Think about viruses can be as big as pox viruses, up to 300 nanometer. HIV up to 150. The smallest autonomously growing virus will be a circovirus, just 15 nanometer.
So what's the smallest HIV particle that's ever been documented?
I think it's something 120 to 150. No, these are fixed morphological entities.
They don't change.
They don't change.
So that's a huge thing in helping you differentiate whether it's HIV or not.
Yeah. The size of a structure is very important to make the diagnosis.
Now, if the particles which are labeled by Bess and his colleagues as being HIV, then the absolutely necessary condition is for the material which was obtained from the infected cultures to have proteins which are not present in the material which was obtained from non-infected cultures.
Right.
But this does not seem to be the case, and in fact, Bess and his colleagues have come with this evidence. They took the proteins from all the three bands they had, from the two infected cultures, or what they called infected cultures, and from the non-infected cultures. And here are the proteins from the non-infected cultures, which they are put in a strip. Here are the proteins from the two infected cultures. As you can see, if you look at this, there is a difference, but the difference between the strips is only quantitative. That is, we have these bands in all the strips, only there is less here. Similarly with all the other proteins. In some of them, in fact, they are exactly the same. So by looking at these pictures, the proteins which existed in the purified material, the HIV purified material, and in the material which was obtained from the non-infected cultures contain the same proteins, which means that none of these cultures contained HIV. As I said, if the cultures which contain HIV, they must have had proteins which are not present in the non-infected cultures. And yet, this is not the case. The proteins, all the proteins are found, which are found in the so-called infected cultures, they are also found in the non-infected cultures. Some of them in smaller quantities, but nonetheless, they are there. The difference may be just because the way the cultures were conducted. The American authors labeled some of the proteins, and the P6, P7, P17, and P24 are labeled as HIV proteins. And these two proteins, that is, proteins which are around 32 and around 41, they were labeled as cellular proteins. No label is put in the proteins which had molecular weight higher than 41. Yet there are many HIV proteins which have molecular weight. Now, why they didn't label them? First, all the proteins which are around 41 and 32 are non-HIV. There cannot be HIV proteins with molecular weight 41 or around 41, and molecular weight of 32 or around 32. Okay. We asked Bess why they labeled these other three proteins as HIV. And he said that they put this label because that's what the reviewer asked them. But they did not have evidence themselves that they were HIV proteins.
He stated that they did not have evidence that they were HIV proteins?
Yes, he stated that they did not obtain themselves, but the reviewer asked them to label them as HIV proteins. And he said the reviewer was right. We labeled them HIV.
Did he specifically state they had no evidence those were HIV proteins?
Personally, they did not have evidence. Yes.
Bess said that?
Yes. In this experiment, they did not obtain evidence. In this experiment, they did not obtain evidence that these proteins were HIV. But they labeled them HIV because the reviewer asked them.